Hello there, and welcome to your first lecture. This talk will serve to introduce us to the field of microbiology, as well as discuss some of the history behind some of the major advances that we had over the last couple of hundred years or so. Microbiology is known as the study of microorganisms or microbes. Microbes come from the Greek word micros for small and organismos for organism. Microorganisms include, but are not, of course, limited to bacteria, fungi, archaea, protozoa, viruses, as well as some algal species. And a large extent of this course is going to focus on bacteria, but we're also going to look at a lot of these others as well, especially in the context of human diseases and infectious diseases. So here you can see a couple of diagrams just showing some bacteria here. We've also got an image of a bacteriophage, which is a type of virus that infects bacteria, and some fungi as well. So all of these are considered to be microorganisms because at least some part of them can only be studied at the micro level, requiring some degree of microscopy in order to look at them up close. Some microbes are considered to be pathogenic, and a pathogenic organism can cause infection and disease. This is obviously a very important thing that is at the forefront of many of our minds, especially because a lot of microbes do cause disease in humans. The other big impact that microbes have in our day-to-day are food spoilage. A lot of the industry that focuses on microbiology has to do with agriculture and also food preparation and food storage for long term. Despite some of these negatives, a lot of bacteria are very beneficial day to day, and they are, of course, a great starting point for many food chains. A lot of the chemical reactions that they actually facilitate will be the baseline foundation that other reactions require in order to proceed. And especially when we consider their roles in things like digestion, for example, they will happen to release factors and different components that organisms couldn't get otherwise. They're also essential for the breakdown or the conversion of toxic materials. In bioremediation and biofuels, they're at the forefront of many research efforts. And when we consider bioremediation, in the case of things like spills or even deposits of long-term pollutants in soils and other things, many microorganisms have metabolic pathways that other organisms simply do not, where they can take these different pollutants and change them into less harmful entities and in turn remediate the environment to a large extent. Nitrogen fixation is something that's really important for plants because a lot of plants have a hard time getting nitrogen naturally from the soil. So they're usually involved in some kind of mutual symbiotic relationship, meaning that both parties benefit with bacteria in order for them to get nitrogen or break it down in a way that's usable for plants. Photosynthesis, we always think about in the context of plants, but some bacteria or microorganisms are capable of photosynthesis on their own, albeit with some different pigmentation rather than chlorophyll. They are really, really important in vitamin production. And in fact, in our own digestive system, there are lots of microorganisms that help to produce different vitamins from our own digestive tract that we would normally not be able to get out of our own food without them. They are also paramount in the food industry and are responsible for making a lot of delicious foods that many of us are able to enjoy, like yogurts, cheeses, vinegars, alcohols, breads, among other things. And with respect to human health, our native flora, or our commensal organisms, protect us from infection. So there's a whole bunch of bacteria that live inside of us natively that are going to compete with different organisms that can be harmful to us or try to infect us. Because let's face it, if you have a home as a microorganism, the last thing that you want to happen to your home is to see it get destroyed by an infectious agent. So what we actually have are bacteria that will fight for you. So there's a whole pile of things that tie directly into immunological responses and how our immune response depends on a lot of these commensal organisms to support us. Now, this will hopefully be a very brief review for you, but a lot of the organisms that we'll be discussing this semester are prokaryotic, and prokaryotes are different from our regular human cells, for example, which are eukaryotic. Eukaryotic cells tend to be larger and more complex. Complexity, of course, relating to the idea that they have more organelles, a higher degree of structure, and compartmentalization, which is not seen at all in prokaryotic cells. Eukaryotes also tend to be larger, generally. There are some awfully large prokaryotes out there, but typically speaking, eukaryotic cells tend to be larger. Prokaryotes, on the other hand, have a simpler morphology, a simpler structure. They lack membrane bound organelles, and especially, most importantly, the distinction is that they lack a nucleus. They do still contain genetic material, but that is not enclosed in a membrane, and of course, is referred to as the nucleoid region. So if you take a look here in this diagram, you can see all of the DNA that's present inside of a bacterium, but instead of being enclosed in that nuclear envelope, it's just free floating in the cytoplasm. I'm hoping by now that you folks might have a pretty good feel for the different cellular components, but if you have not yet sat down and really learned what all of the different parts of cells are, especially for prokaryotes, since that's where our focus is going to be, I cannot recommend enough that you do sit down and familiarize yourselves with the basic anatomy of cells, because when we get later on into the course and start talking about different mechanisms and things that these cells do and undertake on a day-to-day, we're going to be referencing a lot of the cellular structural components, and to have those on the tip of your brain is really going to be helpful for your understanding. There are many types of microorganisms, as we discussed, and a lot of them are differentiated based on what they're actually made up of. How many layers do they have? Do they have an envelope? How many cell membranes do they have? What are their internal components like? So we're just going to take a couple of minutes to go through the different major types of microorganisms and discuss some of the features that they have. And obviously, your job is to understand what the differences between them are. Bacteria are single-celled organisms, and they have a cell wall as well as one or two cell membranes. Their cell walls primarily are made up of peptidoglycan, and you'll notice that a lot of the differences that we're going to see in the next couple of slides come in terms of what is different in cell wall structure. Peptidoglycan is not found in all bacteria, but most bacteria, because there are a couple of groups that have other types of materials, like mycolic acid, for example, that we'll discuss later on, of course. We won't get into this too much now, but bacteria tend to have several distinct shapes. So they can be bacilli or bacillus if it's singular, which is rod-shaped. They can be cocci or coccus singular, which are spherical and spiral-shaped. And you can see the three examples here show bacilli, cocci, and these spiral-shaped. We're going to discuss in detail, more so in the lab component, bacterial arrangements, so how cells tend to group together. Do they group in pairs? Do they group in chains? Do they group in clusters? Etc. There are a lot of different examples of those, but for now we'll leave it there. When they reproduce, they do so through binary fission, which is an asexual form of reproduction, where they just elongate and pinch off to form a new set of cells. In terms of their motility, some exhibit flagellar movement, though there are other mechanisms of bacterial movement as well, and some bacteria that just don't move at all and would be considered to be sessile. Archaea are the other half of the prokaryotic scope. These are also single-celled organisms, but they have a cell wall that is not made up of peptidoglycan. This is the distinguishing factor that makes you either archaea or bacteria. The structure and function of archaea are very similar, but what's really unique about the members of archaea are that none of them actually cause disease in humans. So we don't talk about them very much because they are not particularly relevant to our interests, especially in terms of human health. What they are known for, though, is their ability to live at extremes. So where most bacteria can also live in extreme conditions, archaea go one step further. They can live at the coldest and the hottest places, and also in places where there are not normal, and I put normal in quotes, forms of nutrition. There are some, for example, that thrive in the saltiest of salty places. Extreme halophiles are ones that can live in salt beds, happily, for that matter. So they tend to be extremists compared to other types of microorganisms. We'll talk about them a little bit throughout the course, but most, like I said, of our focus will be on bacteria. Viruses are acellular and, as a result, considered to be non-living. They are extremely small compared to most other microorganisms that we'll talk about, and they require the invasion of a host cell for replication. They are unable to replicate on their own and can't survive outside of a host. They tend to be very simple in their structure. Most of the time, they have a core, which is made of DNA or RNA, but never both. So you can have viruses that are RNA viruses or DNA viruses, but they never contain both. They also usually have a protein coat, and sometimes they also have an envelope, which is made up of a lipid membrane. They come in all shapes and sizes and all types of virulence, meaning how pathogenic they are. They infect a variety of different hosts, they infect a variety of different tissues, and we're going to spend a little bit of time talking about them throughout this semester as well. And finally, here we have some multicellular animal parasites that are obviously eukaryotic, and they aren't microorganisms, but they are of medical importance. So we may spend a little bit of time touching on these topics. They tend to be microscopic at some points in their life cycle stages, and internal parasites are typically called helminths. They are flukes, tapeworms, roundworms, and an abundance of other types as well. And what they tend to be problematic for is replicating to such high numbers in a host that they cause physical blockages. So you can see in this example here where we've got a lot of worms that have packed themselves into some part of a person or an animal and blocked that passageway. This is seen a lot in digestion, especially in countries where there are not a lot of hygienic practices, where worm eggs could be in the foods that are consumed and water that gets consumed. And this is a real problem in those areas of the world. So when we look at the history of microbiology, we have to remember that the field is fairly new on the general scheme of life on Earth. When the world beyond the naked eye was discovered, it was impactful worldwide and completely revolutionized the day-to-day life of people as we knew it. It began with the discovery of the compound light microscope by Zacharias Janssen in approximately 1590. The first compound microscope only magnified things about 9 times, compared to our regular compound microscopes, which can now magnify things up to about 1,000 times. Even this little glimpse into things a little bit closer really blew open people's worlds and started to make them question about what was going on around them. Just as a bit of an aside here too, while I will have dates in a lot of these slides, I'm not interested in those for testing. Mostly, the only thing I would like you to really focus on for tests are the names of the people and what they contributed to the field. So the time frame, it's not super important. In 1664, Robert Hooke was the first person to describe microorganisms after he observed some mold that was growing on leather. And he also first described cells after observing magnified cork. So he could see the little compartments that were formed inside of the cork material. And that was the first time that anybody had ever noticed that compartmentalization. His discovery led to the idea of the cell theory, which states that all living things are composed of cells. And this started to establish a foundation for research and more inquiry into what else existed at magnified levels. At this point, Hooke's microscope was not powerful enough to view microorganisms, and truth be told, even at the limitations of the compound light microscope at 1,000 times, it's still very difficult to tell a lot about microorganisms, but we are able to see them and their general structure and characteristics. So this was just a little bit of a drop in the bucket of what would eventually become a massive field with tons of research and relevant information that would change the world as people knew it at the time. Anton van Leeuwenhoek was the first to observe what he was calling animalcules. He made a lot of drawings of organisms that he observed in rainwater, feces, as well as teeth scrapings. He used to sit in front of his window, you can see the picture here, with his little microscope that he would load up with materials. And you can see this is the glass piece or the lens, and on the end of this little pin, he would load up his samples. And he was able to see different organisms, and these are actually some of his sketches that he produced at the time of things that were there. Maybe they were even moving, and he had no idea what these things were. Van Leeuwenhoek's work was the first glimpse into a previously unknown world, right? If you can imagine just going around your day-to-day and then one day opening up your closet and seeing a portal in the back that led to an entirely new place, It's that degree of bizarre when you consider what these people's day to day was like prior to this discovery. And at the time, a widely accepted theory was spontaneous generation. Back then, folks truly believed that life could arise from a couple of ingredients, for example, like moist earth. Things like toads, snakes, mice, flies could just materialize from nowhere with the right conditions, and this theory was originally proposed by Aristotle. In truth, though, when food is left out over time, obviously putrefaction is going to start to occur as these things rot away, but they're also contaminated with bacteria as well as higher organisms. And there's actually here a recipe for mice, which was written by a fellow in the late 1500s, where if you added together the following in an open pot or barrel, like dirty underwear, a dirty shirt, or some rags, and a few grains of wheat or some wheat bran, if you left it for 21 days, adult mice would appear and be capable of producing more mice, which you would expect. This is not unexpected given our current knowledge of the world. But at the time, people didn't know how these lower organisms, if I can put it that way, came into existence. And spontaneous generation was something that held on for dear life, as we're going to see in the slides that are coming up. In 1668, Francesco Redi set out to disprove spontaneous generation because many people believed that maggots, or fly larvae, could arise from decaying corpses, so that as soon as somebody died, automatically these maggots would be born and could start to consume the flesh. So what he did was he filled 2 jars with decaying meat. He left one of them unsealed and another that he sealed. And what happened over time is that the unsealed flask, of course, had visitors. So flies came and laid their eggs in that meat, and maggots appeared. But in the sample where the flask was sealed, no maggots appeared. Unsurprisingly to us, of course, because no flies were able to access the meat itself. Believers in spontaneous generation, however, rejected that, and they said that maybe it was because there was no fresh air, because how could organisms live if the air inside wasn't fresh or oxygenated or whatever they were thinking at the time? So this was not enough evidence to disprove spontaneous generation to these people. A second experiment was set up with mesh gauze covering a flask, and obviously gauze is permeable, so it would allow for airflow to get into the flask, and once again, no maggots appeared on the meat, proving that they did not spontaneously come out of the meat itself. These results were very firm proof against the existence of spontaneous generation, but people still believed that despite the fact that a large fly maybe couldn't be generated spontaneously, these animalcules that Leeuwenhoek had observed maybe could be. And why couldn't they be produced through spontaneous generation in non-living materials? So the debate raged on. Almost 100 years later, John Needham continued this debate on spontaneous generation when he started boiling things. We all know by now that when we boil things, we essentially kill everything that's living on them, effectively sterilizing them. So what he did was he boiled nutrient fluids like chicken broth or corn broth or other things that had sugars and other nutrient sources in them, and he poured them into covered flasks. Microorganisms were found in these solutions, as you might expect, and Needham claimed that they arose spontaneously from the liquids, even though they had been purified, if you want to think of it that way. So somebody else said, well, maybe the microorganisms that were found in your broths entered through the air. And instead of leaving those open to air, Lazzaro Spallanzini actually came in and repeated the experiment, but actually covered the flasks and prevented air from being able to get in. What he did show was that a sealed flask following heating did not develop microbial growth. Even still, Needham turned around and said, no, the vital force that's necessary for spontaneous generation was destroyed by the heat. Only in this guy's experiment, apparently, though, but was also kept out of the flask because they were sealed. So others argued as well, again, similarly to what we saw with the previous meat experiment, that a lack of air would prevent any organisms from being able to thrive in those situations. So the great debate was ongoing until Louis Pasteur busted on the scene. Louis Pasteur was the person who finally disproved spontaneous generation by explaining that microorganisms that exist in air can contaminate solutions, but that air on its own is not a contaminant and cannot spontaneously generate organisms. This ties directly into Rudolf Virchow's explanation about biogenesis, where all living things can arise only from pre existing living cells. And so this was slowly starting to establish what would eventually be known as what we currently accept today. So, how did Louis Pasteur disprove spontaneous generation? Well, he used a variety of different flasks. He tried some short necked flasks, he tried some long necked flasks, and some curved flasks as well. And what he found was that in a short necked flask, which is shown here in A, that after a few days, even if the solution had been boiled, even if left open to air, they became contaminated with microorganisms. The long necked flask was also left open to air and was also boiled, but because of the curvature, was not able to have things fall directly into the liquid from the open air. Yes, air was still allowed to pass in and out, but things could not contaminate the broth after it had been boiled. And what he was able to show was that no microorganisms actually grew in the flasks and their contents following a couple of days, even though the flasks were exposed to air. The discussion from that point onward was that bacteria are ubiquitous. They live in and on and all around us, and they're airborne as well. Needham's flasks were contaminated with airborne microbes, and air itself did not spontaneously generate contaminants. There was also the onset of mechanisms to keep solutions sterile, and a nice segue into how to keep food from going bad as well, back right before the turn of the last century. Things like heating and other methods that can block access of airborne microorganisms and prevent food from spoiling and prevent, hopefully, people from getting sick. This is also the basis of what eventually became aseptic technique, which is still something that's used in our day-to-day. Wash your hands. Make sure that your food is refrigerated to slow spoilage. Don't leave things open on the counter overnight. There's certain ways that you should be thawing meat if you're a meat eater, of course. Things like human health practices. Also in the laboratory. We often manipulate bacteria using aseptic technique to keep everything nice and sterile. So this was what put all of what we currently know about microbiology and its practice as a field into motion. The period from 1857 to 1914 has been described as the golden age of microbiology, and after these discoveries, things really started to take off. We learned how to culture bacteria and isolate them. We learned a lot about microbial metabolism. We started identifying different organisms that were disease-causing, which was really important, and we'll get to that in a couple of minutes, of course. Surgical techniques were improved, human health was improved, and of course, vaccine development came to be, which again saved a lot of lives. Medical microbiology also very much benefited from Louis Pasteur's discoveries. Diseases were originally thought to arise from supernatural forces. Only bad people got sick. Thieves, criminals, murderers, anybody that did bad things in life was punished by supernatural forces that would actually cause them to get very ill and in some cases die. And when microorganisms were first discovered, some people started making some really important links to disprove that idea that illnesses befall people that are bad. When microorganisms were first linked to fermentation, it was very indicative that they were able to change organic materials. And this kind of paved the way for the discussion around diseases and how microorganisms may make changes in plants and animals. The idea that microorganisms cause disease is known as the germ theory of disease, which is still very much the accepted standard. In 1840, Ignaz Semmelweis showed that disinfecting hands reduced disease transfer, and he was working in a hospital setting as a surgeon and was seeing that surgeons were actually transmitting infections between patients because they were not cleaning their hands in between. Hand washing at that point was not a common practice, and in turn, a lot of people that were having babies at the time were becoming sick from doctors going from one person to the next to the next. In the 1860s, Joseph Lister applied germ theory to medical procedures, and he developed treatment with phenol. Phenol is a compound that is known to kill bacteria, so he started applying it to surgical wounds and getting people to cleanse themselves with phenol prior to embarking on these medical procedures. And instruments were becoming heat sterilized, and the rate of transmission in these settings went down dramatically just from a couple of different changes on how bacteria can be transferred from person to person, being eliminated. The biggest revolution in medical microbiology came from a fellow named Robert Koch. He was the first person to show that bacteria specifically cause disease. He was studying something so dangerous, and if you know anything about anthrax, you'll know that it can be lethal, especially if it's inhaled. So he was looking at anthrax, which is caused by Bacillus anthracis in anthrax-infected cattle and sheep. When he studied these animals a little bit more closely, he was able to observe that these sheep had rod-shaped bacteria in their blood. He was able to isolate these bacteria and culture them outside of the animals on nutrient plates, and then he injected those cultured bacteria into healthy animals. Those animals died. When he isolated blood from the dead animals to compare to the first bacteria, they were identical. And so it was the very first time that he had shown that an infection was transferred to a healthy individual from a sick individual, and was able to show that microorganisms, in this case Bacillus anthracis, were the causal agent of the disease itself. So from this work on anthrax, there was a set of rules that were established to determine if a specific microbe causes a disease. The microorganism has to be present in every case, but absent from healthy organisms. The suspected microorganisms must be isolated and grown in pure culture, meaning that you have to be able to take them out of the melange of microorganisms that may exist and culture them to purity, meaning that there'd be this single entity that you could then inoculate or transfer a new healthy host with. And when that healthy host becomes ill, the same microorganism has to be isolated again from that diseased host. And that conclusively can determine whether a specific microorganism is causing a disease. What came to be known as Koch's postulates required growth of organisms in pure culture, and this is impossible to do in liquid form. So if you think about a broth, for example, think about your beef broth that you make soup with. You add some noodles to it. If you were to have a bunch of different microorganisms growing in there, it would be very difficult to isolate them. What ended up happening was a transfer onto solid media, which started out being gelatin. You might know gelatin from Jell-O, for example. It's the agent that makes Jell-O firm up. Bacteria, unfortunately for these folks, can digest gelatin at room temperature, and so their solid medium became non-usable. Koch had an assistant, and his assistant's wife, Fanny Hess, came up with a plausible answer, which was agar. And agar is still a material that we use today to make agar plates. And these are still used to culture bacteria. It was an ingredient at the time that was used to make jellies, and it was suggested as a base for solid media. Most bacterial species can't digest it. It's very easy to use in a lab setting because it doesn't really melt down or turn into a liquid form until it's heated above 100 degrees, so it has to be boiled. And it also only solidifies when it hits 50 degrees Celsius, which makes it very easy to boil it and then to actually pour it out before it has a chance to harden again. So it was like a dream come true. And what was able to happen from that was isolation on agar. And what were they going to pour the agar into? Well, Richard Petrie, you may recognize the name, came up with the idea of a plate with a lid and a base called the Petri dish. And again, these are things that were discovered way back in time and we're still using to this very day. This is just one of the many cool things that you can do in the lab where you can draw with bacteria. So if you take a loop or another instrument that has been inoculated or has bacterial culture on it and you draw a little picture, when you incubate your plate and it comes back in a couple of days after those cells have had an opportunity to divide, you can make a picture. So there's a lot of agar art out there in the world, which is just a joy to look at. One of many little asides to bring a little bit of joy to your lab work. Now, another really important advancement was the development of early vaccination. In 1796, Edward Jenner noticed that milkmaids that were exposed to cowpox were somehow immune to smallpox. And if you don't know about smallpox, it was a disease that was running rampant through Europe at the time and was killing a lot of people. So what he ended up testing was whether or not somebody could become resistant to an infection by being exposed to a minor, more mild form of the same type of disease-causing agent. And in this case, cowpox is very similar to smallpox, but cowpox does not infect humans. Smallpox does, but cowpox does not. So, in what could be considered a very unethical move, Jenner infected an 8-year-old boy with cowpox by making a scratch in his skin and applying cowpox pustule contents. This first method was called variolation. So it's using live viruses or live organisms that cause disease in one person and deliberately planting them into somebody else to expose them to it. Variolation is not very safe. In fact, it was used a lot before vaccination was first established. So variolation is super outdated. It's super dangerous because we're using live organisms, but it was all that people had at the time to start to develop that idea of immunity. After being exposed to cowpox, then later on, that same poor boy was purposefully infected with smallpox. And amazingly, he showed immunity. He did not get sick. People who got smallpox really had a high chance of dying, and this young man did not get sick following that exposure with the infectious agent. It's very rare to have two viruses like this, like cowpox and smallpox, which are mainly the same, but don't both cause disease in the same set of hosts. So, in turn, in order to have a similar effect, what we ended up relying upon were vaccinations. Vaccinations do something very similar to this, where they take parts of or sections of pathogenic microorganisms that we expose our bodies to form an initial immune response. Vaccines can be made from attenuated organisms, which tend to be less virulent, less potent, like in the case of cowpox to smallpox. They could be parts of microbes, so pieces that we know that our body can form an immune response to once they see it. Microbes that are still intact but that have been killed so that they have no risk of sprouting back to life and causing disease. Or microbial toxins. In the case of things that produce a toxin that has the detrimental effect or causes the disease, you can isolate those and inject those into people. And in every case, what is happening is the body is forming immunity. Once our body becomes exposed to something from outside of itself, which is considered to be foreign or non-self, we have a whole bunch of cells inside of our bodies that come in, recognize that, and fight against it. And that forms what's called immune memory. The next time that we see that organism again, our body's already seen it and knows how to react. By getting a vaccine, what you're doing is allowing for your body to recognize a microorganism that can be disease-causing, but without having the disease-causing organism come into your body and have the risk of getting the disease. So your body still sees the organism for what it is, can still form those recognition patterns. So if you ever do get exposed to the actual thing, you've already got the immunity there. You already know what to look for. Your body already knows how to respond without having to get sick the first time. So the vaccine, in essence, replaces an initial exposure with a disease-causing organism, and it's a much safer mechanism to have that exposure rather than the alternative of getting sick and maybe dying. Knowing that bacteria were responsible for human diseases also led to treatment with chemical substances. Chemotherapeutic agents were starting to be formed, and in the laboratory they can be made as synthetic drugs, or if they're derived from bacteria, fungi, or other living organisms, they are known as antibiotics. Paul Ehrlich in the early 1900s speculated about whether or not there was something like a magic bullet that could be produced that could essentially go out and hunt down and destroy microorganisms without harming the host, and discovered salvarsan, which is a derivative of arsenic that was effective against syphilis. And later on that century, in the late 1930s, sulfonamides, also known as sulfa drugs, had also been synthesized. So we were making very small steps forward in how to destroy microorganisms without also affecting the host. And then came the big hitter. The biggest discovery in terms of antibiotics was by Alexander Fleming. He was growing some bacterial cultures on agar plates, and they became contaminated with mold. When he went to throw them away, though, he actually noticed that there was a zone of inhibition around the culture. So if you look here, this is the bacterial growth, these streaks, and this is the mold culture. And you can see around this, that there is a zone where bacteria cannot grow. And that was very interesting. Alexander Fleming isolated the contents of the agar and discovered penicillin. And this was one of the first of many different antibiotics that was isolated and implemented. We hear a lot these days about antibiotic resistance, which is very true because we've overprescribed and we've overused antibiotics for so many years now that bacteria have developed mechanisms to circumvent these medicines and how they would normally work inside of cells. But that's not to say that we are at the end of antibiotic use, and there are still a lot of different avenues to explore in this realm. But this is something that we still use today when we study how effective a medication is and what dose is required to inhibit bacterial growth. We're going to actually do this exact thing in the laboratory later on in this semester. So I hope that this has kind of set the stage for some exciting advancements in terms of microbiology as a field. And of course, there's still tons of different places for the field to go in today's day and age. We're constantly researching all different sorts of microorganisms. And one very promising area, of course, is recombinant DNA technology, which we will touch on later on, of course. But we're always looking for different things, different ways around these little microorganisms that try to wiggle their way into us and make our lives miserable with human disease. All we want to do is coexist. And there are a lot of different ways that we can adapt in order to continue to coexist happily with these folks in the long term. That about does it for us for today. And if you do have any questions, please feel free to email me anytime at lauraR@nipissingU.ca. Until next time.